traits will tell us which parent had the dominant allele and which parent had the recessive. And we'll be able to tell because of their kids' phenotypic features.
All you hear today makes me better. It makes me.
So instead of looking at the field trip connections that we've made to the biological processes we've been studying this quarter, I chose to look at 3 different processes that relate to the big idea of requirements for life in kind of a different way. I'm going to do this by showing you how the cell cycle, protein synthesis, and genetics all relate to the family that I'm going to be talking about in this video.
Here we see the father on the right along with his parents and brother. And here we see the mother on the right along with her twin sister and her parents. And here's the happy couple whose genes we will be discussing today.
In looking at the cell cycle, this gives us a picture of what would happen during reproduction when when the mother was trying to get ready for the child to be born. So here in the G1 phase, we just have one chromosome from each parent, and then they double in S phase, the most important phase, which is the synthesis and replication phase. And then here in the G2 phase, they're just prepping for division, where here in metaphase, the microtubules will pull each chromosome to different poles, and then during cytokinesis, they will split into a mother-daughter cell, and that is how we have one for the new child. This works much in the same way, except it is for gametic cells, not somatic cells, and it's for the sex cells, and so it goes through the same process, and then in cytokinesis, it splits, also creating 2 daughter cells. Protein synthesis is the next thing we're going to look at. So we start with the DNA here in the nucleus, and it goes through transcription within the nucleus. And so that is how the DNA and the mRNA are created. And so then the RNA goes out, and it is translated, and it happens on the ribosome. And then it heads into the ER, where the tRNA brings in the amino acids. This is for when a mutation would occur here, but we're just going to assume nothing happened. So then the tRNA brings the amino acids in, and this is where it gets folded, either correctly or incorrectly, assuming correctly. And then it will leave the ER, and then it either will get flagged, stored, or it continues on. So, assuming it's correctly made, it continues on to the Golgi, and then it would get sent out. And here is where we don't want mutations. We do not want genetic variation. Otherwise, it could be a disease.
So genetics can be described as a genotype and a phenotype. The genotype is the actual coding for what someone will look like, and the phenotype is what we actually see as the person is created. Now, here we're drawing the Punnett squares for the mom and the dad, and then what their kids would look like. As you can see, the dad had a heterozygous allele for his hair, and so he could be either blonde or brown haired, so blonde earlier in life and then brown. And then the mom, both her parents had brown hair, and so as she grew up, her hair continued to be brown. So for the children, they have a heterozygous chance of being blonde or brown haired, as seen in their picture shown early in the video. Seen in their children, the kids obviously have heterozygous genes because both of them have started out to be blonde-haired. The alleles that they have, they code for the specific traits that they have, and they are pairs of DNA on each chromatin. Mendelian genetics says that you will have complete dominance over one allele, but as we can see with non-Mendelian genetics, this isn't always true, as you can see with these kids, because they're kind of in the middle. They're not blonde or brown hair. They have heterozygous features. So that's how the cell cycle, protein synthesis, and genetics all relate to how these kids were created differently, genetically different, so that they can survive in this world. Thanks for watching.